Next story. Unlocking the secret X factor in zebra finch love songs with I. In a mesmerizing blend of art and science, researchers have uncovered a hidden treasure trove within the melodious world of zebra finch love songs. These enchanting avian melodies may hold the key to understanding the mysterious X factor that captivates female songbirds. Male zebra finches are renowned for their elaborate tunes, a creative showcase of their fitness and genetic prowess. However, the enigma lies in the fact that many of these birds learn only one song in their lifetime. The question arises, how do female birds discern the fittest among them based on a single melody? Enter artificial intelligence. In a groundbreaking study, a team of researchers has employed AI to analyze the intricate nuances of male zebra finch songs. What they discovered left them awe-inspired. Hidden within these melodic masterpieces is an imperceptible X factor, undetectable to the human ear but potentially holding the secret to avian allure. While the exact nature of this mysterious factor remains shrouded in ambiguity, the researchers found that songs containing it were not only more challenging to learn but also exuded a magnetic charm that appealed to female finches. This revelation opens a gateway to unraveling the intricacies of what makes certain bird songs more captivating than others, offering a glimpse into the captivating world of avian courtship. But the wonders don't stop there. From the harmonious melodies of zebra finches, we journey to the realm of skymons, tiny whirlpools of magnetic spin that hold promise for revolutionizing computing technology. By harnessing the computing power of these elusive entities, researchers have unlocked a new frontier in data storage and processing, paving the way for energy-efficient computing systems of the future. As we marvel at the symphony of nature's melodies and the ingenuity of AI-driven discoveries, one thing becomes abundantly clear: the intersection of science and nature continues to illuminate the hidden wonders of our world, offering a tantalizing glimpse into the mysteries that lie just beyond our perception. So, let us embrace the harmonious cadence of zebra finch love songs, the swirling dance of skymons, and the boundless potential of artificial intelligence as we venture deeper into the captivating tapestry of our ever-evolving scientific landscape. Source: Nature article, Nature. Next story. Balancing fish nutrition, optimizing feed and food production. In the vast world of seafood systems, a new study has shed light on the intricate balance between fish used for feed and fish intended for direct human consumption. Small marine creatures like anchovies, herring, and mackerel are bursting with essential nutrients such as vitamins, minerals, and omega-3 fatty acids, making them crucial for both food security and human health. However, the dilemma arises when these nutrient-rich forage fish are predominantly processed into fish meal and oil for aquafeeds instead of being directly consumed by people. But fear not, as recent research published in Nature Food by Willow et al. Has proposed a game-changing solution. By reallocating a portion of forage fish from feed production to human consumption, the nutritional benefits can be maximized on a global scale. The study reveals fascinating insights, showcasing how edible portions of forage fish used in feed actually contain more calcium, iodine, iron, vitamins A and B12, and omega-3 fatty acids compared to the nutrients retained when fed to farmed salmon. Conversely, farmed salmon fillets provide higher levels of zinc and selenium, along with similar quantities of vitamin D, underlining the potential nutritional trade-offs in seafood distribution. The researchers propose a strategic reallocation strategy where 50% of edible forage fish used for food instead of fish oil production could lead to 100% retention of key nutrients like iron and omega-3 fatty acids. This shift could not only address nutrient deficiencies but also generate over 400,000 tons of additional seafood for direct human consumption, presenting a win-win scenario for both nutrition and food supply. However, implementing such a change comes with its challenges, including assessing consumer demand, adapting supply chains, and promoting shifts in dietary habits. The study emphasizes the importance of public-private partnerships, technological innovations, and policy support to drive these transformations in seafood systems. From supermarket campaigns to sustainable byproduct utilization, every effort counts towards optimizing our seafood resources to meet the diverse nutritional needs of populations worldwide. As we navigate the intricate web of fisheries and aquaculture, the key lies in finding the delicate balance between nutritional efficiency and environmental sustainability. With ongoing advancements in alternative sources of nutrients and evolving consumer preferences, the future of seafood systems holds immense potential for creating a more resilient and nourishing food supply chain. So, Next time you savor a piece of salmon or indulge in a seafood feast, remember the intricate journey these marine delicacies undertake from the oceans to your plate, teeming with possibilities for a healthier and more sustainable future. Source: Nature Food. Towards a nutritional balance of fish for feed and fish for food. Nature. Next story. Unleashing the power of protons. In the exciting world of particle accelerators, a recent breakthrough has given us a glimpse into the intricate dance of protons along the line of stability. Imagine a high-stakes game where the players are tiny particles racing through a sophisticated playground of magnetic fields and forces, each move crucial to the success of the game. In a groundbreaking study published in Nature Physics by Hannes Bartosik and colleagues, the existence of stable trajectories in the four-dimensional phase space of protons has been confirmed. These stable regions, known as fixed lines, offer a deeper understanding of complex dynamical systems and hold the potential to revolutionize the design of future accelerators. Particle accelerators are at the forefront of scientific research, driving innovations in fields as diverse as nuclear physics and material science. However, optimizing their performance is no easy feat. The interplay of external forces, self-generated electromagnetic fields, and nonlinear effects poses challenges that demand a precise understanding of particle behavior in phase space. The key to unlocking this mystery lies in the observation of protons as they traverse the accelerator under the influence of linear and nonlinear forces. Quadrupole magnets provide linear focusing, while sextupole magnets introduce n-nonlinearities that can lead to resonances and particle loss. By studying the phase space structure of the proton beams, researchers can gain valuable insights into the dynamics of these intricate systems. One of the most striking findings of this study is the presence of fixed lines in the four-dimensional transverse phase space of protons. These fixed lines, akin to stable orbits in a chaotic sea of motion, offer a rare glimpse into the ordered structure underlying the apparent randomness of particle trajectories. Through meticulous experiments at CERN's superproton synchrotron, Bartosik and his team were able to track the movements of protons along these fixed lines, providing experimental validation of theoretical predictions. By carefully manipulating the proton beams with kicker magnets and beam position monitors, they were able to observe the intricate dance of particles in phase space. While this study represents a significant step forward in accelerator physics, there are still challenges to overcome. Limited experimental setups and potential beam losses present obstacles that researchers must navigate as they seek to further explore the dynamics of particle motion. As we look to the future, the discovery of fixed lines opens up exciting possibilities for enhancing the performance of current accelerators and designing novel facilities for cutting-edge research. From exotic beams for nuclear physics to rare processes in particle physics, the implications of this research are boundless. So, the next time you ponder the mysteries of the universe, remember the tiny protons dancing along the line of stability, revealing secrets that may shape the future of scientific exploration. Source: Nature Physics. Nature. Next story. Unlocking enhanced data storage with magnetic whirlpools. In the realm of data storage, a groundbreaking development has emerged. Magnetic whirlpools offer improved data storage capabilities. These whirlpool-like structures, known as skiamans, are intricate arrangements of spins that can be controlled electrically on a nanometer scale, making them ideal candidates for storing vast amounts of data efficiently and swiftly. Traditionally, data storage involves rotating bits mechanically within the host material, which can be slow and unreliable. However, researchers have now demonstrated an all-electrical method to read and write information by encoding it in a single nanoscale skiamian. This breakthrough, reported by Chen et al. In a recent paper in Nature, paves the way for low-power data storage systems capable of storing massive amounts of data. The key to achieving electrically controlled magnetic states lies in giant magnetoresistance, a phenomenon that induces significant changes in electrical resistance based on the magnetic configuration of a material. By harnessing this effect in magnetic tunnel junctions, researchers were able to engineer a structure that hosts a nanoscale skiamian with high tunnel magnetoresistance. These skiamans, which resemble whirlpools of spins, offer a promising solution to the limitations of traditional data storage methods. What sets this development apart is the ability to switch between magnetic states using an applied voltage, making the process much more energy efficient compared to current-driven methods. By leveraging the interaction between neighboring spins and perpendicular magnetic anisotropy, researchers were able to reliably induce the Scivi-Co state with minimal power consumption, leading to a 1,000-fold increase in energy efficiency for information writing. Furthermore, the potential applications of Scivi-Co-based data storage extend beyond conventional computing. By combining magnetic tunnel junctions and Scivi-Cos, researchers envision applications in brain-inspired computing, where these structures could play roles analogous to synapses and neurons in artificial neural networks. This convergence of computing elements presents exciting prospects for future developments in unconventional computing. While the current study represents a significant advancement in condensed matter physics, challenges remain in achieving field-free methods for optimal writing and developing a unified approach for writing, shifting, and reading Scivi-Cos with a single device. Nonetheless, the realization of a Scivi-Co magnetic tunnel junction opens doors to innovative possibilities in data storage and computing. The journey towards practical Scivi-Co-based data storage devices continues, fueled by the promise of magnetic whirlpools revolutionizing the landscape of data storage and computing. Source: Nature. All electrical Scivi-Ionic magnetic tunnel junction, Nature. Next story. Unlocking the mysteries of blood cell production in bone marrow through advanced microscopy. In a groundbreaking discovery, researchers have unveiled a powerful new microscopy technique that sheds light on the intricacies of blood cell production in the bone marrow of mice. The study, published in Nature by Wu et al., showcases a method for imaging the production of various blood cell types in the bones, revealing a detailed organization of cell lineages in response to stressor like bleeding and infection. Blood cells are constantly regenerated through a process known as hematopoiesis, occurring primarily in the bone marrow of adult mammals. This dynamic process allows the body to adapt to various challenges, such as infections, by producing different types of blood cells. Wu and colleagues have developed a cutting-edge protocol that enables visualization of the most immature hematopoietic stem and progenitor cells, red blood cell production, and the generation of immune cells like dendritic cells and B lymphocytes within the bone marrow. By leveraging a combination of fluorescence microscopy and specific surface markers on bone marrow cells, the researchers uncovered the spatial organization of stem cells and different progenitor cells relative to each other and other anatomical structures within the bone marrow. They observed that blood cell production sites are distinct and non-overlapping, with specific cell populations clustered around small blood vessels. Stem cells were found as solitary entities, while lineage-committed progenitors formed clusters with their daughter cells, showcasing a defined architectural pattern in the bone marrow. Furthermore, the study demonstrated that blood cell production sites are flexible and can adjust their output in response to external stressor, returning to baseline once the stress is resolved. Interestingly, different bones within the skeleton exhibited varying responses to stress, highlighting the intricate regulation of blood cell production throughout the body. While this innovative microscopy technique provides unprecedented insight into bone marrow anatomy and hematopoiesis, the widespread application of this technology may face challenges due to the complex requirements of fluorescently labeled antibodies and advanced imaging equipment. Future research directions may involve enhancing image analysis software and integrating molecular profiling methods to deepen our understanding of disease progression and immune responses within the bone marrow. This groundbreaking discovery opens up new avenues for studying blood cell production dynamics and its role in health and disease, offering a fresh perspective on the biological complexities of the bone marrow environment. Source: Nature. Next story. Unraveling intricate movements resulting from robot interactions. In the realm of robotics, a groundbreaking discovery has shaken the very foundations of our understanding of motion. Picture a row of simple mechanical rotors, each connected by elastic bands and sitting atop an electric motor. These are the building blocks of a revolutionary system known as a robotic metamaterial, designed by a team led by Veenstra et al. Unlike traditional robots that adhere to the laws of conservation of energy and linear mechanics, this robotic metamaterial defies our expectations. It introduces a tantalizing element of non-reciprocity, where the motion of neighboring rotors becomes entangled in a dance of opposing directions. This intricate interplay of forces gives rise to a phenomenon known as solitons, wave-like structures that move in unexpected ways, akin to ripples in a vast cosmic pond. But the real magic happens when magnetic fields are brought into the mix, creating pairs of stable states within the system. Picture a delicate balance, where each rotor can rest either tilted to the left or to the right. When nudged, These rotors trigger a chain reaction, causing a mesmerizing ripple effect as boundaries between left and right tilted domains traverse the metamaterial. What sets this discovery apart is its self-sustaining nature. The energy required to propel these solitons forward is not injected from an external source but is harnessed from within the very essence of the robotic units themselves. This self-sustenance opens up a realm of possibilities for applications in processing and filtering mechanical signals, hinting at a future where robots not only mimic nature but enhance it. As we peer into this world of non-linearities and topological defects, we catch a fleeting glimpse of mechanical logic taking shape before our eyes. The journey doesn't stop at solitons, it beckons us towards a realm where even more complex interactions await exploration. Veenstra and colleagues have not just unveiled a robotic marvel but have opened a gateway to a universe where robots and metamaterials orchestrate a symphony of motion beyond our wildest dreams. So, as we marvel at the solitons traversing the robotic metamaterial, let us remember that within these seemingly simple rotors lie the seeds of a mechanical revolution, where strange physics dance hand in hand with cutting-edge robotics. Source: Nature article, Nature, next story. Decoding the intricate messages in birdsongs. In the intricate world of birdsong, researchers have embarked on a transformative journey to unravel the complexity of avian communication through an innovative lens. The melodious tunes that male songbirds sing during courtship play a crucial role in attracting potential mates. While traditional methods of analyzing birdsong focused on basic parameters like pitch and duration, a pioneering study by Alam et al. Took a novel approach, harnessing the power of machine learning to delve deeper into the nuances of these enchanting melodies. By utilizing cutting-edge computational tools, Alam and colleagues introduced a holistic method to measure song complexity by evaluating the spectral distinctiveness of individual syllables in a bird's song. In their study, the researchers focused on zebra finches, recording thousands of song repetitions and processing the data using a sophisticated deep learning syllable detection algorithm. Each syllable was transformed into an image representing its sound frequencies over time, which was then visualized using a data visualization technique called UMAP. The UMAP visualization unveiled a mesmerizing map of sounds, where each unique syllable formed distinct clusters based on its spectral characteristics. By measuring the shortest path length between these clusters, the researchers could quantify the spectral distinctiveness of a bird's song. Remarkably, female zebra finches showed a preference for songs with syllables that were more spread out in the UMAP visualization, indicating a correlation between path length and perceived song quality. Moreover, the study revealed intriguing insights into the learning behavior of juvenile birds. Juveniles learning songs with longer path lengths face challenges, suggesting that songs with higher path lengths might serve as indicators of high-quality mates. This discovery not only sheds light on the intricate nature of avian communication but also opens doors to a deeper understanding of the evolutionary significance of birdsong in mate selection. While the study heralds a new era in the analysis of birdsong, questions remain about the interpretational nuances of UMAP projections and their implications for understanding animal behavior. Nonetheless, the integration of machine learning in studying birdsong represents a significant leap forward, offering a more nuanced and comprehensive perspective on the captivating world of avian communication. As we venture further into this captivating realm of birdsong exploration, the fusion of technology and biology promises to unveil even more secrets hidden within the melodic symphonies of our feathered friends. Source: Nature article, Birds convey complex signals in simple songs, Nature, next story. Unveiling the immunological memory of brain astrocyte cells. In a groundbreaking discovery that challenges conventional wisdom about the brain's immune response, a team of researchers led by Li et al. Has found that astrocyte cells in the central nervous system possess immune memory. These star-shaped cells, known for their crucial role in maintaining brain health, have been shown to exhibit molecular memories of specific responses to injury and disease, resulting in modified inflammatory signaling that sheds light on autoimmune disorders. The study, published in the prestigious journal Nature, unveils a fascinating mechanism through which astrocytes can acquire epigenetic immune memory, enhancing their pro-inflammatory reactions to specific stimuli. By investigating the impact of initial pro-inflammatory triggers on astrocyte responses to subsequent challenges, the researchers observed a significant increase in the expression of pro-inflammatory genes, indicating a true augmentation of the cell's inflammatory signaling capacity. Moreover, The researchers explored the relevance of this immune memory in the context of autoimmune diseases like multiple sclerosis using a mouse model of experimental autoimmune encephalomyelitis, E. Astrocytes derived from EAE-afflicted mice displayed heightened pro-inflammatory responses compared to healthy astrocytes, suggesting a priming effect that amplifies inflammatory signaling. Further investigations revealed that this enhanced response is mediated by epigenetic modifications involving histone acetylation, controlled by the HATP300 and the enzyme ACLY. Deletion of these key regulators from astrocytes attenuated disease symptoms in the EAE model, pointing to a direct link between astrocyte epigenetic memory and pathological inflammatory responses. This discovery challenges the traditional view of the brain's immune system and underscores the pivotal role that astrocytes play in regulating CNS inflammation and autoimmunity. The findings not only offer new insights into the complex interplay between brain cells and immune responses but also open up avenues for potential therapeutic interventions targeting astrocyte immune memory in various CNS disorders. As we delve deeper into the intricate mechanisms governing brain health and immunity, this research highlights the dynamic nature of astrocyte function and the far-reaching implications of immune memory in neurological disorders. By unraveling the mysteries of astrocyte immune memory, scientists are paving the way for innovative treatments and a deeper understanding of the intricate workings of the brain's immune system. Source: Nature. Next story. Unveiling the key bacterial subspecies linked to colon cancer. In the world of cancer research, a groundbreaking study has uncovered new insights into the bacterial subspecies potentially driving colon cancer. Fusobacterium nucleatum, a bacterium typically found in the oral cavity, has been linked to a variety of health issues, including sepsis and gynecological infections. However, its connection to colon cancer was first established in 2012 through genome sequencing of tumors, sparking a wave of investigation into its role in this deadly disease. Recent research published in Nature by Zepeda Rivera and colleagues has delved deeper into the relationship between F. nucleatum and colon cancer. What sets this study apart is the identification of key subspecies of F. nucleatum, FN anomalies, FN polymorphum, FN vincentii, and FN sensu stricto, that are directly associated with colon cancer development. These subspecies form cancer-promoting biofilms in colorectal tumors, indicating a significant role in disease progression. The study further narrows down the potential culprit to FN anomalies, specifically focusing on two distinct clades within this subspecies. Surprisingly, only one of these clades, known as FNAC2, was found to be highly prevalent in colorectal tumors, highlighting its unique role in promoting cancer. Through comprehensive genomic analyses, the researchers uncovered specific genetic and metabolic signatures that distinguish FNAC2 from its counterpart, shedding light on its potential mechanisms of action. While animal studies yielded mixed results in terms of cancer-promoting potential, the human data solidified the association between F. nucleatum, particularly FNAC2, and poor clinical outcomes in colorectal cancer patients. These findings underscore the importance of further investigating how this bacterium contributes to tumor progression and metastasis, paving the way for future diagnostic and therapeutic strategies. Overall, the study by Zepeda Rivera al. represents a significant leap forward in our understanding of the complex interplay between bacteria and cancer development. By honing in on specific bacterial subspecies like FNAC2, researchers are moving closer to unraveling the intricate mechanisms underlying colorectal cancer. As the field of cancer microbiology continues to evolve, these findings hold promise for improving patient outcomes and advancing precision medicine in the fight against this deadly disease. Source: Nature. Next story. Unraveling chaos. Abel Prize awarded to mathematician for taming randomness. In the realm of mathematics, where unpredictability reigns supreme, a groundbreaking genius has emerged, shaking the very foundations of randomness. Michel Talleyrand, a mastermind in the field of probability theory and functional analysis, has been crowned with the prestigious 2024 Abel Prize for his remarkable contributions to mathematical physics and statistics. Picture this: a river's water level fluctuating wildly, influenced by myriad uncontrollable factors like rain, wind, and temperature. This chaotic dance of randomness was once an enigma until Talleyrand unveiled his ingenious inequalities, a set of formulas that breathe order into the chaos. His work is not mere calculations, it's an exquisite piece of art, painting a picture of predictability in the face of uncertainty. But Talleyrand's achievements extend far beyond mere equations. He played a pivotal role in solving a perplexing puzzle posed by theoretical physicist Giorgio Parisi, a conundrum involving the intricate structure of spin glasses. Parisi's initial attempts were met with skepticism, as his methods seemed like sorcery to the mathematical community. However, Talleyrand, armed with his brilliant mind, unraveled the mystery and paved the way for Parisi's Nobel Prize in Physics in 2001. Talleyrand's journey to mathematical stardom is as unconventional as his methods. Born in France with a vision impairment, he faced academic challenges but emerged victorious through sheer determination and a renewed focus after a near-blindness scare. His unconventional path led him to the pinnacle of mathematical research, where he continues to inspire and challenge others to push the boundaries of what is deemed possible. In a world where randomness once reigned supreme, Michel Talleyrand stands as a beacon of order and predictability. His work has not only revolutionized the field of mathematics but has also reshaped our understanding of the universe's intricate dance of chance and determinism. As we celebrate his remarkable achievements, we are reminded that even in the face of chaos, there lies a hidden order waiting to be unraveled by the daring minds of visionaries like Michel Talleyrand. Source: Nature. Next story. Unveiling COVID-19's impact on the brain. In the ever-evolving landscape of the COVID-19 pandemic, the impact on the brain has emerged as a compelling area of study, shedding light on the complex neurological symptoms associated with the disease. From loss of smell to cognitive difficulties, the virus has been found to affect the brain in various ways, presenting researchers with a challenging puzzle to solve. Recent research has revealed intriguing clues into the mechanisms behind these neurological manifestations. While some studies suggest a direct invasion of brain cells by the SARS-CoV-2 virus, others point to a systemic inflammatory response as a key player in driving cognitive symptoms. The balance between these two theories has sparked intense scientific debate, fueling a quest for a comprehensive understanding of COVID-19's impact on the brain. One study conducted by neuropathologist Helena Radbruch and her team at Charité Berlin University Medicine found evidence of heightened immune activity in specific brain regions of individuals who succumbed to COVID-19. This neuroinflammation was particularly pronounced in areas responsible for functions like smell and breathing, hinting at a potential link between systemic inflammation and brain-related symptoms. In parallel, research led by neuroscientist Matthew Campbell and neurologist Colin Docherty at Trinity College Dublin uncovered a concerning phenomenon: a compromised blood-brain barrier in individuals with long COVID and cognitive impairment. This breach in the brain's protective barrier could pave the way for inflammatory molecules to infiltrate the brain, potentially contributing to conditions like brain fog. However, the plot thickens with studies suggesting a more direct role of the virus in brain dysfunction. Investigations by neuroscientist Shuaibing Chen of Cornell University demonstrated that SARS-CoV-2 can infect specific neurons, triggering a cascade of cellular events associated with aging and inflammation. These findings underscore the intricate interplay between viral invasion and host immune responses within the brain. Amidst the scientific whirlwind, the quest for therapeutic interventions and biomarkers intensifies. From exploring the potential of existing medications like metformin to dissecting the intricate web of factors driving COVID-19-induced brain damage, researchers are fervently pursuing avenues to alleviate the cognitive burden of the disease. As the puzzle pieces of COVID-19's impact on the brain gradually fall into place, the journey to unraveling its full complexity continues. Each new discovery brings us closer to deciphering the enigma of neurological symptoms in the context of this global health crisis, offering hope for improved treatments and interventions in the future. Source: Nature. Next story. Unveiling the chaos, stars that devour planets in the Milky Way. In the vast expanse of the Milky Way, a hidden chaos has been unveiled by a group of stellar detectives, shedding light on the tumultuous lives of stars and planets in our galaxy. Imagine a star, millions of years old, calmly sitting in space, only to reveal its secret appetite for planetary bodies. Yes, you read that right, stars feasting on planets. In a groundbreaking study published in Nature, a team of astronomers has uncovered 7 stars that have indulged in a planetary feast, challenging the conventional belief that mature solar systems are usually stable. These stars, in their middle age, have left a trail of celestial breadcrumbs that hint at a history of planet ingestion, a process that was thought to occur mainly in a star's early life. This discovery opens a new chapter in our understanding of the dynamic interactions between stars and planets. Astronomers used a cosmic twin study technique to identify sun-like stars in our galactic neighborhood that exhibited signs of planet-eating behavior. By analyzing the composition of these stars and their paired siblings, the researchers were able to pinpoint the outliers that had devoured a rocky world. What makes this finding even more intriguing is that the planets met their demise relatively recently, within the last few 100 million years. Some were engulfed as their atmospheres eroded, while others were captured as rogue planets on a collision course with their stellar fate. This revelation challenges the notion of a serene and stable universe, showcasing the dynamic and chaotic nature of celestial bodies in the Milky Way. As we ponder the implications of these planet-eating stars, one question lingers: what other cosmic secrets lie hidden in the depths of our galaxy? Are there more stars with a taste for planetary delicacies waiting to be discovered? Only time and further exploration will reveal the full extent of the hidden chaos that permeates the Milky Way. So, the next time you gaze up at the night sky, remember that beyond the twinkling stars and distant planets lies a universe full of surprises and mysteries, waiting to be unveiled by the relentless curiosity of science. Source: Nature. Next story. The impact of Putin's election victory on Russian science. In a recent turn of events, Russian President Vladimir Putin has been re-elected for a fifth term, securing a whopping 87% of the votes. The international community has criticized the election, citing issues with fairness and freedom. But what does this mean for Russian science? Many scientists fear that another 6 years under Putin's rule will lead to further isolation for Russian science. With the ongoing conflict in Ukraine, Russia's global standing in the scientific community has taken a hit. Collaborations with Western countries have dwindled, pushing Russian researchers towards partnerships with countries like India and China. Despite Putin's public support for research, the scientific landscape in Russia is facing challenges. Sanctions and political oppression have led to a brain drain, with thousands of researchers leaving the country. Internal discussions at institutions like CERN have also resulted in the termination of ties with Russian research institutions. The impact of sanctions on Russian science has been significant, affecting even the most productive scientists. The isolation has disrupted collaborations in fields like Arctic research and medical sciences, with implications for the country's expertise and progress in these areas. While scientists in exile work on alternative visions for Russian science, those who remain in the country face supply chain disruptions and personal risks. Nationalist ideologies are on the rise, influencing research and higher education in Russia. As the global community navigates the aftermath of the election, there is a call for revitalizing Russian research and bringing it back into the international fold. Despite the challenges ahead, there is a sense of resilience among scientists who believe in working towards a brighter future for Russian science. As the world watches the implications of Putin's re-election unfold, the future of Russian science remains uncertain. The resilience and determination of scientists both within and outside the country may pave the way for a new chapter in Russian research. Source: Nature. What Putin's election win means for Russian science. Nature. Next story. Unlocking cell biology, fascinating protein moments captured on film. In the world of science, where mysteries await to be unraveled, a groundbreaking technique is changing the game. Imagine witnessing the intricate dance of proteins within cells, a sight previously unseen. Enter time-resolved cryo-electron microscopy, a cutting-edge tool that is transforming our understanding of the microscopic motors and devices that drive life itself. Picture this: myosin, the protein powerhouse responsible for muscle contractions, has long been scrutinized by scientists. However, a crucial moment, the pre-stroke state where myosin readies itself for action, remained elusive. That is until now. Researchers have captured this fleeting state, lasting mere milliseconds in living cells, using time-resolved cryo-EM. These movies of proteins in action are revolutionizing biology, offering unprecedented insights into the inner workings of cellular machinery. Leading the charge is a team of structural biologists who are turning still snapshots of molecular processes into dynamic motion pictures. With just 2 frames, they have confirmed decades-old theories about myosin's choreography. This is just the beginning. From cell signaling systems to gene editing tools like CRISPR-Cas9, scientists are poised to explore a plethora of molecular machines with newfound clarity. But how does time-resolved cryo-EM work its magic? By flash-freezing protein samples in liquid ethane and imaging them with powerful electron beams, researchers can capture snapshots of individual molecules in action. These snapshots are then transformed into structural maps, revealing the dynamic nature of biomolecules that was previously hidden from view. With this technology at their fingertips, researchers are delving into a realm of biology that was once beyond reach. From studying the activation of G-protein-coupled receptors to unraveling the mysteries of viral entry molecules, the possibilities are endless. Every frame of these molecular movies holds the key to understanding complex biological processes at a level of detail never seen before. While challenges remain, such as the need for specialized equipment and precise timing, the potential of time-resolved cryo-EM is undeniable. As researchers refine their techniques and push the boundaries of what is possible, a new era of biological discovery dawns. So, next time you marvel at the complexity of life at the molecular level, remember that hidden within each protein's motion is a story waiting to be told. Through the lens of time-resolved cryo-EM, the secrets of the cell are coming into focus like never before. Source: Nature. Next story. Groundbreaking. Human receives first ever pig liver transplant, lasts 10 days. In a groundbreaking feat of medical innovation, a team of surgeons in China has successfully transplanted a pig liver into a 50-year-old man, marking a significant milestone in the field of xenotransplantation. The genetically modified miniature pig liver remained functional inside the recipient for an impressive 10 days before being removed. The procedure was conducted at Zijing Hospital of the Air Force Medical University in Xi'an, with the aim of exploring the potential of using pig organs to bridge the gap for patients in need of liver transplants. This achievement opens up new possibilities for individuals with liver failure, offering a temporary detox solution while they await human donors. The pig liver, which had been genetically engineered for compatibility, exhibited normal color and texture throughout the transplantation period. Surprisingly, the liver was able to secrete bile, a vital sign of its functionality. This successful experiment has instilled hope in the medical community and laid the foundation for future advancements in xenotransplantation. The surgeons involved in this pioneering surgery hope to gain further insights into the safety and effectiveness of pig organ transplants. By studying the immune response, infection risks, and overall liver function, they aim to optimize the process for future clinical applications. While this remarkable achievement represents a significant leap forward, challenges remain in utilizing pig organs as long-term replacements for human organs. The liver, with its complex functions, may serve as a temporary fix for individuals requiring detoxification or waiting for a suitable human donor. However, the ultimate goal is to develop sustainable solutions for organ transplantation that can benefit a broader range of patients. With ongoing research and collaboration among experts in the field, the potential for xenotransplantation to revolutionize the treatment of organ failure is becoming increasingly promising. As scientists continue to push the boundaries of medical science, the prospects for enhancing and saving lives through innovative procedures are brighter than ever. This successful pig liver transplant sets the stage for future advancements in xenotransplantation, offering hope to patients in need of life-saving procedures. Source: Nature. Next story. Exploring lessons from a fictional hospital ransomware attack on a remote learning platform. In the ever-evolving landscape of healthcare, a new concept has emerged: the hospital-at-home platform. This innovative approach integrates digital technologies like telemedicine, wearables, and sensors to provide hospital-level care to patients in the comfort of their homes. The benefits are immense: reduced risks of hospital-acquired infections, increased patient satisfaction, and potential cost savings. However, a recent simulated scenario brings to light a crucial aspect that cannot be ignored: cybersecurity. Imagine a ransomware attack crippling the ward-at-home platform, locking down servers, and compromising patient data. The consequences are dire: inability to deliver care, patient deaths, and a loss of trust in the healthcare system. The scenario unfolds with the launch of the ward-at-home service, boasting of AI-driven monitoring and real-time health alerts. But as the platform grows and accommodates more patients, it becomes a target for cyber threats. The attack on a Sunday night leaves the operations center scrambling to respond, unable to communicate with patients and staff. The key question raised is whether such a cybersecurity incident can be classified as a black swan event, an unpredictable catastrophe with severe repercussions. The answer lies in the preparation and mitigation strategies put in place. The need for thorough risk assessment, robust defense mechanisms, and continuous monitoring is emphasized to safeguard healthcare systems from such attacks. As the healthcare sector increasingly relies on digital technologies, the vulnerabilities to cyber threats become more apparent. It's imperative to prioritize cybersecurity in healthcare infrastructure to prevent major public health emergencies. Policies and guidelines recognizing the criticality of cybersecurity in medicine are essential to protect patients and maintain the integrity of healthcare systems. In conclusion, the imagined ransomware attack on the hospital-at-home platform serves as a wake-up call to the healthcare industry. It underscores the urgent need for proactive measures to secure digital healthcare systems and ensure the safety and well-being of patients. Source: Nature.